This is TWIS. This Week in Science, episode number 928, recorded on Wednesday, May 31st, 2023. You oughter be listening to This Week in Science. Hi, everybody. I'm Blair Bazderich, and today we will fill your head with spiders, gene editing, and curious kids. But first...
Thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. For the past many decades, science has been hot on the heels of various treatments, cures, and therapeutic solutions for a long list of diseases. Gaining knowledge through observations of correlations, aided by genetic sequencing data, biochemical analysis, and a sustained, dedicated, iterative effort to find solutions. Most of the time, those solutions remain elusive, even to the best of scientific efforts. Just skim through the concluding statements of research papers. Over the past many decades, research into cancer, Alzheimer's, autism, Parkinson's, or stem cells? What you will find at the ends of hundreds of thousands of these papers is a passing of the torch, a hopeful plea to the fates dressed in professional caveats, something along the lines of, these findings may provide a pathway for future research to further explore the potential causative nature of correlations observed in our study, and suggesting that eventually somebody might stumble upon a key to unlock the door to potential clinical treatments. That is how science functions. Small steps of definitive knowledge gains, eventually leading to treatments, techniques, and cures. Science is the ultimate long-form story, which is why we offer it to you in long-form installments of This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Blair.
And happy World Otter Day to you, Justin.
Is it that time again?
It's that
My
time
goodness, the year
again.
goes by so quickly, doesn't it?
Yes, yes.
World Otter
And good
Day.
science to everyone listening or watching out there. Thank you for joining us for This Week in Science. We have a great show ahead. Kiki will be joining us later on. She has stories about curious kids, sweetener damage, and useful AI. What do you have, Justin?
I have, so the whole thing in the disclaimer about iterative steps and, a future path to a thing that maybe one day, I've got actually two stories that have reached the door, key in hand, of unlocking the future of scientific therapies, or of clinical therapies through science. As well as, of course, I had to bring a Neanderthal story, right? That's a given. And the thing I didn't know about sex education.
Oh, interesting.
Yeah.
Is that your fault or your teacher's fault? Well, we'll find out.
Well, it's West Virginia's fault, actually.
Okay.
Yeah, we'll get to it.
Got it. Okay, and I brought seagulls and spiders. Just like when you see Neanderthal news, you have to bring Neanderthal stories. I saw some more funnel web spiders in the news, so I had to bring them. So they're joining us in the animal corner. And then I also have a quick story about silent zoos. And no, I'm not talking about the animals. I'm talking about the humans.
Oh, that would be cool.
Yeah. As we jump into the show, I wanted to remind you that you can subscribe to TWIS, This Week in Science, as a podcast on your favorite podcast platform, YouTube or Facebook. Anywhere you see our video or audio, please subscribe. And that will bring you TWIS each and every time we release a new episode. Just search for This Week in Science or visit twis.org. Are you ready to start talking about science, Justin?
Yes.
Great. What do you have?
Oh, yeah, I can start, with
You gotta start. Kiki's out here.
Okay, so let's see. Where do we want to start? Okay, this one. This is a crazy one. So this is researchers at the Hebrew University of Jerusalem and the University of Haifa in Israel. They have discovered that production of nitric oxide in the brain correlates with autistic symptoms. It's something that they had previously discovered in some research. And so they decided to kind of delve into that a little bit further. So nitric oxide is a multifunctional signaling molecule in the brain. So it's doing all sorts of neurotransmitter work and regulation. It's involved in cell survival. It's also involved in the plasticity of the brain and how the vasculature of the brain works. It's kind of important to a lot of things. And one of the things they noticed is that there were elevated levels in mice that were genetically altered to have autism. So they decided they were going to delve into this a little further in this follow-up study. First thing they did is they took regular mice and they basically increased the levels of nitric oxide. They gave them a drug that leads to an increase of nitric oxide production. And they saw some significant reduction in cortical dendritic spine density, which is something that you see in autism patients and mice who have been genetically altered to have autism. They then went the other way around, the complete opposite, and they took genetically altered mice to have autism, which is just a knockout of one or two genes. And they used nitric oxide inhibiting drugs. And what they found on those mice is that it actually reversed the biochemical signals of the dendritic spine architecture, biochemically at least. It was reversing the effects of autism. They then did a third round to see what the behavioral changes were. And so they had these mice who were again modified to have autistic-like behaviors and with the same types of genes that are altered in humans. Put them through the paces. They showed this, lack of interest in novelty versus familiar objects, lack of social interaction, these sorts of things. They again did the inhibition of nitric oxide, and the behavior changed. So, this is in mice. However, in discovering a mechanism, first it's a signal of, a biochemical signal that is different that was observed. But then they managed to actually recreate it in experimentation and reverse it. So they have found not only a mechanism involved, right? A causative, perhaps. It's downstream from the genes. So the genes are doing stuff that's going to, this pulls a lever over here. This changes the amount of gene expression of this, which is an editing or a regulating factor, and all these things sort of play into a cascade of nitric oxide going up. So that's why mechanism versus what it actually, finding a gene only tells you so much.
Right.
That, okay, we have these genes associated. Now there's this whole mechanistic cascade of things that happen afterwards. That's the really, in this day and age, the difficult part to even figure out. But here they've identified one, did an increase and a inhibition of it, and saw dramatic effects. So they may have found a mechanism towards, again, this is still in mice, you got to do all the human trials and everything else later, but to find a single target, single possible drug target that can, in vivo, while alive, do an alteration of behavior, of biochemical outcome. Very huge. And so look for this. This is, I mean, I'm not saying this can reverse autism,
Right.
but it did in mice that were genetically altered to have it, with the same genes that are different in humans. It really comes down to two genes that are related to.
Well, but it didn't. So it didn't reverse autism though, because to your point, it didn't edit the genes. It just treated the symptoms. It changed the behavior. So it didn't exactly reverse it, because if you stopped treatment, in theory, everything would revert back, right?
Correct. And it's behavior in mice, so you don't know how much that translates. Like even if it was a perfect analog for humans in this test, and they did a clinical trial and it turned out it was the perfect analog, how far ranging those behavioral, cognitive, and everything else effects are in a human compared to a mouse is hard to predict what that relationship is, but still pretty big deal.
Yes.
Pretty big deal.
So there's, we often try to trace things like this to the source, which is your previous point, right? Like if you could trace this all the way back to the genes, and you could alter genes so that this isn't an issue, that would be
and they know the
option
genes.
Number one. That would be what you'd want to do, right?
And they're working on, they did the experimentation across two different genes that are associated with the vast majority of autism cases. And so by knocking those genes out is how they get to autistic behavior in these mice, and some of the biochemical dendritic architecture that is similar.
Right. So you're treating symptoms. You're not kind of reverting the biology back. So you're going one step back. You're going one step back. You're one step back from, I'd say, symptom. Because you are attacking a causative, which is an overexpression of the nitric oxide, meaning that it is curative with a continued treatment. Right. But that's exactly it. It's a continued treatment. So it's
for mice. Yes. Still, it's for mice.
Yes.
We're not humans yet.
It's a good development. I'm not saying it's not a good development. I'm just saying I think science is going to continue to move kind of backwards.
If we had the ability to edit genes in a living being, then we could, Right. Oh wait, but that's going to be my second story.
Associated with all sorts of things.
Yeah.
Well, go ahead. What's your second story?
So my second story is they did gene editing in live mice.
Perfect. What for?
So this one, they as a proof of concept, they altered risk genes that normally lead to coronary disease and Alzheimer's. Mm-hmm. Proof of concept. Why not? As long as we're in there. But this is a Harvard University researchers. They've just improved the gene editing process for the prime editing method, which is something that came out a few years ago. One of the advantages of prime editing is that the system does not cause double-stranded breaks. It sort of nicks one side. It might do it on both sides, but it still doesn't open up the whole whole DNA all at once. And that approach minimizes, when it comes back together, there's less deletions or additions, little goofy things that sneak in when you're doing something like CRISPR. And it's very targeted. If you know anything about genomics and you see a description with nice images and video and everything, it shouldn't work. It's kind of wonky looking. They grabbed the Cas9 and they added this whole extra structure to it, and the whole thing is sort of wonky and leads to this flap of half a strand of DNA flapping in the wind at the end of it. That's just sort of, the repair system apparently takes care of. Anyway, they have this thing. The problem is targeting. It works great in a lab when you're applying it to a cell, and you can edit that cell, and that's great. But now we're talking about what would we need to do to put it into a living body. So the current method of doing gene targeting, is through something called adeno-associated virus vectors. So these are little, basically hollowed out viruses that can be outfitted to attach to specific parts of the chromosome, and they can carry a small payload of DNA with them. So the payload capacity, though, is too small for the prime editing mechanism. The solution? Break the prime editor into 2 parts and send it off on 2 viruses. So now you have a part A and a part B that have to self-assemble when they get to the right place. And they did it. So that's pretty amazing. Self-assembly within proximity of the gene that they're supposed to edit. And then, so that was the huge workaround, was just one in a series of other obstacles. So by doing this, I guess there was a little bit of that wonky system with the flaps and the things attached to each other that I was trying to explain that shouldn't work. Got a little bit wonkier. So they reinforced the RNA scaffolding. They switched promoters. They then ran out of room, so they added a 3rd virus. So now they have a part A, part B, and part C that are recombining. They optimized this with a reverse transcriptase enzyme. Modified Cas9 with a mutation, got everything working, and then downsized it by picking off parts that they actually ended up not needing. Got it back down to 2. Now we're back to part A and part B. All of this, the results were, in a mouse model, the prime editing was able to do 42% efficiency of gene editing in the mouse brain, 46% in the liver, 11% in the heart. This is the highest levels of gene editing in a living creature that has ever been done.
And it's less than 50%.
Not 50% accuracy. This is there was no off target. There was no off target or indels, which is the deletions or additions, but it reached 42% saturation, I suppose you could call.
Right. So if you're trying to eliminate the incorrect sequence,
right,
in favor of the correct sequence,
mm-hmm.
How much do you need to alleviate symptoms?
Right. So what you end up with is, so for instance, you have a cancer risk gene. Okay. Now, what a cancer risk gene is it's usually a gene that's that's repairing DNA. It's the superhero gene's expression, creating things that are fixing DNA when damage takes place. So when those are not what we might call wild type or the preferred type, when they have a little variant and things aren't working right, and the repairman comes out there, but I forgot my wrench. And, I don't have a wrench because my DNA has this weird, you can't fix things. That's the damage that takes place that then can become cancer. So, if you have the genes that are going to allow that cancer to form because of this broken type or altered type DNA, it can happen in any of that tissue that needs repair. If 40% of those tissues have been fixed, you've just reduced the chances of the risk factor that you may have for having, cancer or whatever the disease is. In this case, they actually worked on Alzheimer's for the brain, which they got to like 40-something%. That lowers your risk, I would guess, by that much, at least connected to the genes. So this is, I think, one of the biggest, we've had a couple big ones. There was a couple weeks ago, the stem cells that weren't getting rejected and were staying in the macaque monkeys. Now we have live gene editing in mice. we have a potential reversal of Alzheimer's. This is a good time. Science is getting to the end of those paths through the valley of fields and all this sort of thing. So again, this was as a proof of concept of just getting the model prototyped. They managed this. So
well, I'm excited for the human trials.
Yeah. Because then that thing that just got discovered that can, mitigate the symptoms by taking out some of the mechanisms, you can go back one layer and say, why don't we just fix the genes?
You can just do
Why don't
both.
we just put the genes? Yeah, we'll just do both. Why not?
Perfect. Well, that'll be very exciting. It's like, I feel like that's decades of us talking about it, finally getting somewhere.
Patrick says, can we stop curing mice and start curing humans? It's mice first. It's because mice are the ones that are actually running
then
the
monkeys,
planet. I don't know if you've
humans.
noticed. I don't know if you noticed. Mice actually run planet Earth. So it's why you take the oxygen mask when they fall, the parent, the one in charge, needs to put it on first to make sure everybody else is taken care of. At least that's what the mice tell me.
Right. Yes. Did the mice tell you about the silent zoo, Justin?
So at first I thought, what a great idea. And then I immediately thought, you know who's at the zoo more than anybody?
Who?
Children. There's more children at the zoo than anywhere else on the planet. They're loud.
Let's follow me on this kind of thought experiment. Silent in terms of your behavior. I am not asking about other people around you. Okay. So how did you feel after going to a zoo or an aquarium or a museum, when you went with a bunch of people maybe you hadn't seen in a while? You spent the whole time talking. What kind of emotions did you have? How did you feel about your time in that space?
Well, okay, so now I gotta think. When have I ever gone to a museum or a zoo to meet up with people?
I do it all the time. I don't know what you're talking about, but
like almost never. But
you've never met up with like your friends that also have kids at a zoo and gone, "Hey, how's it going? Let's walk around the zoo together." You've never done that?
Maybe.
Okay.
No,
well,
not really.
I'm guessing a lot of people listening have done that. But when I do that, I can say that when I leave that space, I had fun. Yeah. I certainly caught up with my friends. I'm happy I spent the day there. I enjoyed it. But now I want you to picture how you feel after you've gone to a museum or a zoo or an aquarium. Perhaps by yourself. Self-paced yourself through the space and not had anybody to talk to about what you were looking at. How did you feel when you left?
So if it was an art museum, I actually got to enjoy it.
Interesting.
Yeah. I actually walked out of there knowing what I had seen. Or some memory of having seen things. Mm-hmm. Versus if I take kids to an art museum, I don't see anything. I'm just watching hands to make sure they're not touching things or that nobody's running away.
Yeah. So this is a study from University of Exeter, and they wanted to look at silent zoo tours. So they ran some special silent events at Paignton and Bristol zoos. These are all in England, as part of a wider project on the auditory culture of zoos. So there were other people around who were talking, but these people, this particular subset, was put through this silent event. So they were not allowed to talk to each other, and importantly, they also had to turn off their devices and not engage.
Oh, that's tough.
Yeah. So visitors participating were better able to focus, concentrate, and meditate on specific animals and their behavior, which sometimes fostered feelings of intimacy with and attachment to particular zoo animals. The silence helped them pay more attention, as you were talking about, meaning that they got things out of their visits that they wouldn't have ordinarily. They were allowed to choose their own paths around the zoo, move at their own pace, but they weren't allowed to talk to anybody. And then they had these focus groups afterwards to discuss their experience. Their silence did affect the pace of their visits. They went around slower and they took more time to think and concentrate. This also meant that they stopped more often and there were general periods of physical stillness that were not present when they were talking. It produced feelings of stress relief, tranquility, and peacefulness. And many participants reported that they felt their silence had affected the behavior of the zoo animals around them. So they said that the animals seemed more comfortable with their presence. They were more ready to come close to them than around noisier individuals. And that silence was considered by some visitors to establish points of connection between themselves and the animal. So they were able to kind of spend time thinking about what they had in common with that individual and really spend time connecting to the individual animals in the zoo. So I'm not saying this is groundbreaking or shocking.
You know,
but I think it's an important recognition of what we use these spaces for. Yeah. If you think about a space as a social space, or you think about a space as a meditative space or a space to connect, those are very different goals. Neither is better than the other. But I think it's a really interesting idea that I'm going to take to heart. I know when we would travel all the time for TWIS and I would go, I went to the Philadelphia Zoo. I had a long time before my flight after both of you flew out. And I walked around the zoo by myself for like 4 hours. And it was beautiful. I got to kind of stop and read the signs I wanted to read. And I got to look at the things I wanted to look at and not look at the things I didn't want to look at.
I was just thinking about those signs because I go to the zoo once a week. It's walking distance from here. I know there's signs all over talking about the animals, saying stuff. And I was just thinking, I haven't read any of those because I can't take my eyes off them long enough, or keep my eyes on them long enough because I got this kid I got to keep watching. Right. Who I'm talking to, or I'm like going, hey, look over. No, you're looking at that. You're actually looking at a pigeon. There's a tiger if you turn your head. You know.
So, I mean, I might argue it's a little too soon to do this with your little one, but when he's a little older, it might be interesting to experiment. Okay, for the next 20 minutes, we're not going to talk. Here starts the timer and see what happens, right? Like, I understand you can't tell a young child not to talk at all for an entire day at the zoo. That's crazy. And also it kind of defeats some of the purpose. Some of the purpose of a zoo for a younger child is to say, like, what do you notice? What do you see? Do you have any questions about this animal? So verbal communication is an important part of learning and experiencing a zoo also. But I think that ultimately this is an interesting reminder, especially if you visit the same location over and over, or you're looking for a new reason to go to the zoo, or you're going somewhere to center yourself and spend some time with yourself. Turn off your phone and don't talk and see how it goes. I think it's a really interesting social experiment, and I would love the idea of including more opportunities to engage in these spaces, maybe holding more silent tours for the general public for people to sign up for. I think that'd be so awesome.
Yeah, and also I think it would make society better. But that might just be my attitude towards hearing other people talk.
Hi, Kiki.
Hi, we are obviously having the This Week in Zoo silent hour at the moment as I get in here. Yes.
So to close out my story on silence in zoos, silence visits, researchers say, can help visitors to develop new perspectives on zoos and their animals, and can also help researchers imagine future possibilities for the auditory culture of zoos. And as I alluded to, I would expand this to other cultural institutions. I think it is also relevant to those experiences. So anyway, I thought that was a really cool way to look at it. I hadn't really thought about it, and I'm going to keep that in mind for my future visits as well.
Yeah. I think that the library rules. You know, if you're gonna talk, you don't need to yell.
Yeah, just absolutely.
If you're in public, you know, we don't need to hear your family's or your friends' conversations if we're standing 20 feet away from you. We don't care.
Yeah.
And
there's a
someplace
way
like
to
the
handle
San
it.
Francisco Zoo, they also have a playground. So there's an
Yes,
entire
exactly.
play
Yeah.
area where if it's loud time, take the kids
If there's
there.
kids involved, forget about it. It's
Even the Japanese gardens, they're the very nice meditative places to go, but you take kids and suddenly they're not so quiet anymore.
Oh my goodness.
Yeah, where are we?
We are still in the quick stories, Kiki,
Yes.
and we just finished all of ours. So if you would like to
My
introduce
floor
turn
us
is yours.
to some of yours, that would be amazing.
I am going to just take this and run with it then.
Yeah.
Woo! Hey, everybody. I had a story out of a whole bunch of big institutions, actually. Gladstone Institutes out of the San Francisco Bay Area, the Broad Institute of MIT, Harvard, the Dana-Farber Cancer Institute. They all worked together to try and figure out whether or not they could get machine learning, which we all are talking about these days. And we are here on this show also are known for talking about the negative impacts of artificial intelligence, machine learning, large data sets. This particular study, though, is trying to figure out how machine learning can help us understand how large networks of genes work together to control how cells work, and also how mutations in those gene networks can lead to disease or disruption. And so they basically gave lots of genetic information to a machine learning algorithm. And then they were like, hey, so, you know, we know there's a bunch of data on which genes get disrupted and kind of some networks that work together. It's like in the cancer gene family, you have certain genes that we know, like BRCA genes and others that are known to be involved in causing cancer or allowing those cancerous signals to be perpetuated. Well, this research, which was just published in Nature, really created a model for understanding how genes in our DNA interact. And the machine learning algorithm went so far as to not only identify genes in particular networks, say related to cancer, that we already knew were involved, but also identify genes that we had never thought or recognized as being involved in certain cancers before. And then to test what was going on, they're like, okay, so let's knock out these genes in a couple of mice and see what happens. And lo and behold, their cancer was not as they were more likely to get cancer, less likely to be treated, and it was really fascinating insights. So machine
Wow.
learning, yeah. One good side here is that now we can really start to understand how all the genes interact, genes that are close together within chromosomes, further apart in chromosomes. And because of the way that machine learning picks up on things in ways that we have not been able to in these massive data sets, it can lead us to maybe drug targets or, other understandings of disease.
So let me ask this. I'm sorry if I missed this.
Yeah.
But so this machine learning system,
Mm-hmm.
did it figure this out by looking at patterns and making an extrapolation? Or did it figure this out by just processing data and making conclusions from the data? Because I feel like it's slightly different.
That is a nuance for sure. So this was they pre-trained their AI in something that it's a machine learning technique that they're calling transfer learning. And so they pre-trained their AI on a data set, they pre-trained what their algorithm is called, which is called gene former. And then they wanted to see the knowledge that they were pre-trained on. So genes that are known to be involved in heart disease, genes that are known to be involved in cancers, things that we already knew. Then they took it and tried to transfer that data that the algorithm had been trained on in a new area that it had not been applied to previously. So it's as if it's a bit extrapolating. It would be
Wow.
taking the information that it was trained on and then basically taking that information, that knowledge
Right.
set, and transferring it to a different modality or a different platform.
Right.
Um,
So it's a lot more than an
yeah.
in and out machine where it
Yeah.
was able to do computations or assessments that we couldn't do just because we don't have the computational power. It was actually drawing conclusions
Yeah.
based on patterns it learned.
Right. And so that's the interesting
Okay.
and why the idea of a gene network is important, and why
Mhm.
there are certain genes that they're close together on chromosomes, and so they always act in concert, or because they have other genes or non-coding DNA that's interspersed in between, maybe we don't notice that they're involved, but they are involved. Like, we know there are thousands of genes involved in intelligence, but we don't know which ones.
Right.
And
Right.
so this is potentially, the kind of machine learning that could start to suss out some of the patterns that we have not been able to see previously.
Wow. That's awesome.
Yes. So it's making predictions is what it's doing.
And we do now know some of the genes involved in cognition. We have some of them. We have some. We have a pretty good list of all these things growing. The thing is, I find interesting about these is that there are multiple pathways. Like once you get start to get when you're talking about the network of genes that are involved in any one process, it's not like there's just if it could only be just one. And sometimes it is, but a lot of times there's lots of elements involved. So when we're talking about the BCRA, right?
Yeah.
Breast cancer related gene. This is the gene that is keeping you from getting breast cancer, keeping most people from getting breast cancer. It is
there are other genes involved, though.
Yes.
Yes.
It's a repair gene. Now, that repair gene is also calling up the supply depot for hey, I need more duct tape. I need you to send duct tape over here. And if the duct tape factory is broken, well then it doesn't matter that you've got the perfect, repair gene. It can't get the supplies. And then the duct tape gene is like, okay, yeah, I've got the duct tape here, no problem. I'll send the driver to deliver it to you. Now, if there's a problem with just the delivery driver, again, you can't make the repair, right? So there is all these interdependencies. And so the big correlations
one can go wrong, a number of things can go wrong. It's just all sorts of different places. And that's what and that's why it's so hard to figure out which parts
big correlations
of the network.
we've seen through, here's one list, here's the other list, let's compare. Aha. Okay. Yeah. Right? But then here's the same list, and some of these people are getting cancer and some of these just aren't. And then now we're starting to talk about what the machine learning has revealed, all those other players that we didn't see in our two comparable lists that are showing up in the hits and not showing up in the misses. And I think it's going
to be really interesting to see how this kind of predictive machine learning works again in comparison to and also in conjunction with our big data set analyses that we've done that have allowed us sometimes to falsely identify certain genes that because of the way that the correlations work within our big data set analyses. So it's going to be really interesting to see if we can use these various strategies together and see how it all works. But, this is no silver bullet. It's not just going to be like, yeah, now we know all the networks, but it's one of the tools that's going to help us get there. Mm-hmm. Predictions, and then we have to test them and we have to make sure it's good, but
and then we can edit them.
Yes.
Then we edit them and fix them.
Yes. And that's the cool part.
Perfect.
And let's see next one I wanted to talk about was slime, because I don't know, and it has nothing to do with watching Nickelodeon as a child. Researchers at the University of Massachusetts Amherst have published in the open journal iScience their work on microbial slime. Do you know how when you go into your sink or your shower and it hasn't been cleaned in a while and you like in the drain area and you're like, oh, it's kind of slimy. I should really clean that. That's not just the microbial mat that's being formed. It's the microbes actually producing a slime.
Okay.
Which is also known as an extracellular matrix.
Uh-huh.
And you're saying you can clean this?
Clean it?
Oh, don't go to Justin's house.
I learn something every day. I thought that's just how sinks were.
So these research anyway, we're trying to figure out, okay, what good is this extracellular matrix? Well, of course, it's going to help to support the bacterial societies that are formed, these biofilms. It's going to have all sorts of functions, and it does play a role in things like antibiotic resistance, clogging tubes in medical devices. There's also, if we can figure out how this extracellular matrix is involved in the survival of various species of microbes, maybe we could learn how to, live healthier, safer lives. Anyway, Barry Goodell.
If your medical tubes are getting clogged by microbial slime, I think you have a hospital problem.
Could be.
Not a micro problem. I think
could be a bit.
Take that back to a different layer of, because they should know not to be trying to even. That's, by the way, this is one of the rare instances. 2, maybe 3 things on the planet that I'm okay being wasteful. Medical devices should all be one use. Disposable diapers. We just lost Blair. I was going to tell her, don't fall for any sort of like, oh yeah, we should do eco diapers. Now, that's one of those things you sacrifice. You say, as a society, we agree to pollute disposable diapers. I think that's an important one. And then the other one, of course, is the plastics that we use in research. There's certain areas where you just don't want to involve extracellular matrix.
Well, yes, you don't want to involve these matrices in there. So how do we get rid of them? Well, a researcher at the university, Barry Goodell at the University of Massachusetts Amherst, says, I've always been interested in the microbial extracellular matrix. And people usually consider it inert and just protective, but he's discovered it functions as a conduit to allow nutrients and enzymes into and out of microbial cells. And so its stickiness makes it so that microbes can't clump together. Or its lack of stickiness will make it so that they can't clump together. So the question is, how do they do it? And they determined that there's a secret ingredient, oxalic acid. And this is an organic acid that's naturally found. And these microbes, many of them, seem to be using this oxalic acid as they combine it with a carbohydrate outer layer. And that creates this microbial slime, the sticky jelly extracellular matrix. And so the microbes themselves can use different amounts of oxalic acid to minimize or exacerbate the amount of stickiness of this matrix, and that will allow more molecules in or out of the cells, so they'll be able to absorb more nutrients or fewer nutrients. But the researchers say they're hypothesizing that there's also the excretion of molecules through the extracellular matrix that also allows them to attack things or digest them externally. And so Goodell thinks There seems to be a sweet spot where the microbe can control its acidic level to adapt to its specific environment by keeping some larger molecules like enzymes out while letting smaller molecules readily pass through the extracellular matrix. And this tuning, if we can control how things like oxalic acid are being controlled by the microbes, then that might influence whether or not we can protect various diseases. And, it's natural, quote unquote.
Yeah, it kind of occurs to me that this is just also the microbial solution to being stuck on a two-dimensional plane.
Sticking yourself to it? Or
No, it's also creating three-dimensional travel within that microbial universe so that you can be separate from your neighbor and stacked up on top and up into the side or whatever, without all having to be surface bound all the time. It allows a little bit of a microbial city to form.
A little bit like a little microbial city. It's like a little city, but it can, the wall, that slimy wall, is what either prevents intruders or allows the attack to occur. How thick do you create the castle walls? How full, how dense is your moat? How much slime do you have in your moat? And my first interesting kind of story that we should all be considering is the amount of artificial sweeteners that we're using in our foods we love our sugar. Yum, sugar. People are like, no, sugar's bad for you. Use an artificial sweetener.
So we're talking about sugar alternatives.
This is an alternative, and specifically this study out of North Carolina State University, published this week, relates to Splenda, otherwise known as sucralose. Researchers at North Carolina State University previously showed that there are lots of fat-soluble compounds that get released when sucralose is ingested and digested. And Susan Schiffman, who is one of the authors on this study, says our new work establishes that sucralose 6-acetate is genotoxic. Not just genotoxic. It goes in and it destroys your genes. Any cell it gets into, it destroys your genes. We found that trace amounts of sucralose 6-acetate can be found in off-the-shelf sucralose even before it's consumed and metabolized. This sucralose 6-acetate is a metabolite of sucralose digestion, but it's already in there in trace amounts in the sucralose in your Splenda packet. And the quantity that could be safe for us per day, we're blowing way past what the European Food Safety Authority has already said is okay. The researcher says, to put this in context, the European Food Safety Authority has a threshold of concern of 0.15 micrograms per person per day. And the work that they've done suggests that trace amounts of the sucralose 6-acetate in a single daily sucralose sweetened drink exceed that threshold. And that's not accounting for any of the metabolites produced after the consumption of that drink.
So is this bad for you, like dangerous, like in the come on, that's not good for you sort of thing? Or is it like this needs to be removed from the shelves because it's toxic? Where are we? So
we are in the petri dish testing stage where they exposed blood cells, and those blood cells were damaged. They also exposed gut tissue to the sucralose 6-acetate, and they determined that when the gut epithelial tissues were exposed to it, they found that it causes the tight junctions between the cells in your gut to become less tight. And so molecules that you would normally poop out end up going
In
into
the
your
blood.
bloodstream. And so
No.
yes, so it causes what's considered leaky gut syndrome.
No,
So
not that.
this hasn't been shown specifically in people according to their studies, but there's evidence of health effects of sucralose ingestion. But the work is what they're saying. It's raising a whole bunch of concerns, especially additionally, the not just causing leaky gut, the cells that were exposed themselves, they had a change in activity related to oxidative stress, inflammation, and carcinogenicity. So increase
You know, we were
in
just talking about gene
cancer
networks.
problems.
Do you know we were just talking about gene networks and how
Yes.
all these little things are independent
And this is-
on each other?
That's-
And then this one's going to just come in there and be like,
Yeah.
yeah, knocking things off the shelf, messing up the phone lines.
Yeah. I
destroying the
think this is
communication.
funny because when I feel like when Splenda first hit the scene, the rumors were that it gave you cancer. That's what you always heard.
The
right?
rumors, yes.
Yes. There's no science, but just rumors that it gave you cancer. And this is not that. Also bad though.
Exactly. Yes, and so it's mounting evidence is so they have
Yikes.
it's reason for continued research. It is reason for concern. It is not, oh, this is doing this for sure for certain, but it is opening up a lot of the information that would suggest that we really need to be considering it very carefully.
So perhaps
Yeah.
just assuming you're not a diabetic or have other things going on, perhaps just a tiny bit of natural sugars might be better.
So the really tough thing here is the number one indicator. You could almost diagnose health issues off of weight. Obesity is associated with all causes of death, higher than anything else that you would look at metabolically. It is associated with human death at a phenomenal scale. Obesity.
And it's expected to become a bigger problem as the decades
So
go
then
forward. Yeah.
you have people who are trying to make wise choices
Yes.
by using alternatives to avoid obesity. And if those are also killing people or have the potential, right? I'm not disclaimer. I didn't read the study. Disclaimer. But if those are also going to put people's health in greater jeopardy, is that more diplomatic? Then that's not a solution. Then it's exacerbating a problem for people who already have basically a health condition.
And so it just goes to show that there is, a lot out there that if we are making alternatives, there needs to be more work done ahead of time to determine their safety for consumption and, the likelihood. And maybe we can use machine learning to help us with all the chemicals that we want to be making safer as we get old, as we move forward into
and
our
I am
future.
a big fan. I'm a big fan of food science in general.
Yeah.
However, I would like to point out to everyone, there are foods that people have eaten for tens of thousands of years that are available in the grocery aisle of the grocery store. I sometimes question why we need all these alternative foods when we have food
It all
already.
Well,
Like,
I'm just going to put it all down to big sugar.
It's also food deserts, it's availability, it's disproportionate costs of fresh food that aren't really appropriate. There's all sorts of problems going on.
Availability.
I feel like you can live cheaper on fresh produce than just about anything else.
So I probably have at least 10 studies I could send you about that. We'll talk about it another time.
We'll talk about it, yeah. When we have the studies to back up the conversation. Absolutely. We hope that we're feeding your brain well here at This Week in Science. Thank you so much for joining us for this episode of our weekly show. If you are enjoying This Week in Science, please make sure to tell a friend today and take the time to head over to TWIS.org and click on that Patreon link, because we can't do this without you. We really appreciate your support. Gonna come on back now with some, what is it? That time in the show that we call, Blair's Animal Corner.
With Blair.
Yeah!
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
For the second week in a row, I have brought you current MVPs of the Animal Corner, funnel web spiders.
I just don't understand why you're doing this to yourself or to us.
Oh man, it's just so
All right, bring it.
exciting. I don't know what's going on with these funnel web spider researchers, but it's all dropping and there's exciting news. So this is from James Cook University. They were looking at the venom, which is the thing that makes them famous. They are considered the most venomous spider in the world. And they have found, bottom line, that the venom of some of these funnel web spiders vary depending on circumstances that those spiders are going through. So this is important because not only are they the most venomous, but those venoms are the most complex of those studied in the natural world. They might be related, those two facts, I would assume. And so they're valued for medical research because a lot of medicine starts out as venom. It's where we get the inspiration for them. And so there's lots of research going on with funnel web spiders looking for therapeutics and natural bioinsecticides that could be in the venom. So they're looking for molecules in these venoms that could be used for human health, for pest management, for all these sorts of things. So lots of funnel web spider research going on. And so in this study, they examined the venom produced by different species under different conditions. They looked at 4 different species of funnel web spiders. They looked at the border ranges spiders, darling downs spiders, southern tree-dwelling spiders, and Sydney funnel web spiders. They were all subjected to different tests, like being prodded with tweezers or puffed with air. So basically, they were trying to stress them out. They mapped the behavior so they could kind of see how stressed out they were, and they measured their heart rate with a laser monitor.
What?
So that they could get a proxy for metabolic rate. I didn't know we could do this with spiders. That's amazing.
And it's much better than going in for actually check the pulse rate, with the most venomous spider.
Well, through the exoskeleton. Pretty difficult to do.
Yeah.
So,
oh, come on. Just flip over. I'm going to just, palpate your
oh no, I broke your leg off. Oops. So then they collected the venom and analyzed it with a mass spectrometer. They found that certain spiders had variations in their venom based on factors such as defensiveness and heart rate. Specifically with the border ranges funnel web, the expression of some of their venom components were associated with heart rate and defensiveness. So the specific components in the venom changed depending on their, mental state, excitedness. Difficult to exactly use human words on what was going on with them, but they were in a different state of mind, I guess. They had a different focus in that moment. The other species didn't demonstrate this, so this does seem to be a species-specific response, which is wild. So the use of the venom and the display of aggressive behaviors both have metabolic costs. So the question is, does one not compensate, but pull resources from the other? Is it possible that spiders use behavioral strategies to compensate for costs? So basically, They increase their metabolic rate when they use venoms. They reduce their movement when facing a threat, so they're not overclocking their metabolic rate. Is there some sort of push-pull going on with their behavior and their venom related to threats?
That's fascinating. Yeah, that's a fascinating question. Is it?
Yeah.
I mean, and okay, so we know, like with scorpions, that they have their anatomy is different. So those that have less venom, they have bigger clippers for pinching. Their pinchers are bigger, right? And those that have more venom have the little pinchers. You got to watch out for that tail, right? So that's anatomical, but is there also a metabolic difference?
Right.
Yeah.
And that's not only anatomical, but it's constant for that individual.
Right.
This change from 10 minutes ago to now. Their venom can be a different concentration or have different metabolic properties based on how they're feeling in that moment. So this obviously could impact further exploration and understanding of the ecological role of venom, the study on animals with venom. If this is something that isn't just these spiders, if venom can change depending on kind of excitedness, metabolic rate, whatever you want to call it, is venom research going on in a state where animals are relaxed or where they are excited? And does that impact the venom research? When something is an anti-venom, do you need a different anti-venom depending on if the animal was surprised and it kind of happened all at once, or if they were amped up and stressed out and finally struck? Is there a metabolic difference? Is there a venom difference? This is an interesting idea. Yeah, that venom could have kind of different states of matter in an individual depending on how they're feeling.
Especially like you said, these have very complex venom profiles. A lot of different molecules going to work in there to create the end substance.
Yeah, to kill their lunch.
Or to protect themselves.
Of course.
Or as we found out last week, not to be used at all during the springtime when they're mating.
Yes, absolutely. Just hoist the ladies in the air. Speaking of lunch, have you ever been... Hey, I'm going to go back to the zoo because this is my personal experience. Have you ever been at the zoo and you bought yourself a delicious lunch? Maybe some French fries.
At a zoo?
Yes.
Yeah, that's about the most delicious lunch at a zoo.
It depends on the zoo. Some zoos have delicious food, some do not. But you bought yourself some French fries, maybe some very questionable French fries. But in the end, as you exit the cafe, a seagull swoops down and steals your lunch. Has this ever happened to you or someone you know?
I just saw this happen at the Copenhagen Zoo with a woman who had bought an ice cream cone kind of thing. And had just turned around with this completely perfect ice cream cone, and a seagull was on the roof of it, swooped down, took a bite, and flew off. And then she just had this look of like, what just happened? That's like, just out of the blue seagull attack. So yes, I see it at the zoo all the very aggressive seagulls.
The beach. It can also happen at the beach. Absolutely.
The beach is where I have experienced it personally. I even had a seagull come up so close it tried to swallow my big toe.
No, thank you. Not delicious.
And
right on
UC Davis
on
campus. UC Davis campus. The squirrels will steal your lunch.
Well, this study's not about squirrels. It's about seagulls. But Kiki, I have a question. When that seagull decided to eat your toe, were you eating toes at that time?
I was not eating toes, but we did have food, and we
did it look like toes?
It may have been a zoo French fry.
So the reason I ask is
there's finger sandwiches. I don't remember.
A recent study on the herring gull, which is a type of gull in the UK, wanted to look at how these gulls associate what food is good with what food is not, if they pay attention to what humans are eating and if that impacts their behavior, and how they decide how kind of bold to be in their food stealing decisions.
I really want to know where the seagull that tried to eat my toe.
Well, let's explore that, shall we?
Let's explore this a bit.
Yes, absolutely. So, previous research has already shown that urban herring gulls adapt their foraging behavior to human activity patterns. So they will come for food when humans are around because humans bring food. They increase their attention towards a person in possession of food. We've all been there. And that they prefer food that has been touched by a person compared to food that has not. So they know that we like the delicious stuff. So we already know all that. But this new research was looking to see if birds could not only track objects handled by humans, but if they could compare the objects in the environment with those being manipulated by a person. So can they say, that person over there is eating a hot dog? That trash can? I think I see a hot dog. Must be delicious. So can they make the connection between humans eat hot dogs, there's a hot dog, I'm going to eat that, that looks like food.
Okay.
So they placed 2 crisp packets, it's a study from the UK, crisp packets of
Potato
different
chips.
colors. Yes, thank you very much for translating. Of different colors on the ground, a few meters, see, again, UK, in front of single or small groups of gulls on Brighton Beach. Researchers sat on the sand, held a 3rd crisp packet that matched the color of one of the packets on the ground, and recorded the gulls' response. So their research was to sit at the beach and eat potato chips. You're welcome. Master students, what a great gig. They, of course, also observed the gulls. And as they hypothesized, they would choose the crisp packet that matched the color of the one they were eating 95% of the time.
Wow.
Amazing.
Yes.
I
So
want what you had.
Exactly. So this
Yeah.
suggests that the gulls possess the ability to identify and compare objects within their surroundings. That human is holding a blue crisp packet. I'm going to go for the blue crisps. They're probably more delicious. The gulls seem to observe the foraging choices of others, specifically people in this case, and use the information they obtain to decide what to eat. Now, where this is weird is that herring gulls have not evolved alongside humans. In fact, as far as researchers can tell, their urbanization began 80 years ago.
That's recent. Oh my gosh.
Yes!
Whoa.
So this isn't a coevolution. This is an extended period of exposure to humans. This most likely is the result of a broader, more general behavioral repertoire. They have been able to watch and mimic foraging behaviors of other animals, and now they can then apply that to us and our junk food, essentially. So this shows seagulls as, unlike we usually like to think about them, smart, versatile, adaptable.
Yeah.
They have been able to adapt these foraging behaviors that they have to urban environments and use really impressive observational skills and behavioral flexibility to do that. What's also interesting about this study is that they looked at the ages of these guys. So you can always tell a juvenile gull because they are brown. And then when they are adults, they turn, yes, they're hiding from predators. When they're adults, they are kind of the white and gray variations that you're used to seeing in the various kind of gulls. Around 86% of the pecks to the chip bags came from adults, even though the population that they observed was only about 46% adults. So the grand majority of these guys the adults far outweighed the juveniles. So does it mean that stealing food requires a certain level of boldness? Or does it mean that the adults just were pushing the younger birds out of the way for the chips?
Or is it that it takes a while to learn the behavior? And
Yes, absolutely.
Yeah, that it's just not, I mean, yes, a certain amount of boldness, and perhaps it is, I mean, I don't know enough about herring gull behavior specifically, but the boldness of a bird is more likely to be, I imagine, increased as it gets older,
Right.
and knows more and survives better. You know, if I've
It
survived
knows
this
what's
long,
safe and what's not. Yeah,
yeah,
absolutely.
So I'm also kind of wondering here now, though, if this is taking a cue from humans, or if this is competition strategy. The idea being, oh, humans like the blue ones. Well, I better eat the blue one first, because I don't want them coming after my blue one. Because these chips laid out on the beach, those are mine.
right.
That human over there is eating other blue ones. I got to get to that one first, because
That's
otherwise
the
the
good
human
one.
will want it.
Yes. Well, the human
Right, so
already does
that's
want
what it
it.
eats.
Yeah.
The other ones I can eat anytime, because the human doesn't. I don't know that the human would even eat that.
Hmm.
So
that's interesting. I will say, based on what I read, it's not a question of which packet they pecked first. It's a question of which packet they stole.
Yeah. Oh, but
So
still, you would,
I don't see anything here about any individuals returning.
Well, because that wasn't part of their study. It doesn't. I mean, eventually, tell me those 3 other chips stayed there at the end of the day, and then I'll be like, then those birds starve without seeing humans. I don't believe it, but I do believe that they would see. They would see a competitive thing for that food type and go after it. That makes more sense to me than like, wonder what the humans eat. I should try that too. I feel like it's more of a competition strategy, because you would see that in competition with other birds.
Yeah, and that's what I would say, is that it's not necessarily an either or. It could be a both. That it could be, if there are many, if these are social animals, these birds, they they have their chicks usually in large colonies. There's lots of birds. They are competitive with each other and other birds. They're probably watching, paying attention to a lot of things. And so there is that cue of another animal is eating this particular one.
Mm-hmm.
I'm going to go after it and get it before that animal comes back and gets it, or before another bird comes to get it, because we now know that one is good. So there could be learning, there could be boldness, there could be competition involved, and thank goodness my toes weren't there this time.
So let me also remind you, though, that again, these guys have only been around humans for about 80 years.
Just wild.
Humans,
it takes about 4 years for them.
We don't usually have a bag of crisps that are poisoned and a bag of crisps that are not, which is why I think it's easy for you to kind of draw that conclusion that it's a competition thing. But if you're observing animals, you could eat a snail that is poisoned or a snail that is not. You could eat a frog that is poisoned or a frog that is not. You could eat a plant that is toxic or a plant that is not. And so I also think there is a huge evolutionary benefit to seeing a particular animal is eating a particular type of thing and going,
dying.
That's not going to kill me.
Yeah, no, I get that. But I mean, also because of the lack of association with humans and all of this, I would be more interested in seeing an experiment where they had worms, bread, and chips, and then something that's pretending to eat the worms maybe, and then they go after the worms first. I feel like it would be you would want to compete with what the resource that was under competition then, knowing you had those other ones that did not seem to be under competition.
Yes,
yet.
The only problem is you'd have to find 3 foods that are of equal value.
Of equal desirability and nutritional value.
If gulls love worms, your experiment is not going to work.
Exactly. This is why it would be difficult. This is why we could just use different colored chips and see if they did. Oh, wait, that's what they did.
That's what they just did. Oh, my goodness.
Well, anyway, point being, the understanding the reason that urbanized animals go after certain types of food can be helpful for us coexisting peacefully. And it can be helpful for educating the public, especially because just saying don't feed the birds is not an effective strategy if they are going to go for whatever you're eating. And so that's something to consider. But I also think this is a good opportunity to look into the intelligence of some animals that we have once again underestimated, called stupid, assumed to have a low IQ because they do things that we think are dopey, but really they have a pretty complex social understanding to be able to do what they do. So
I'm with you right there. Yeah. This is I think gulls are fascinating, and they are predators, they are scavengers, they are social. There's so much going on there. And we went, oh, they're just birds. It's just whatever.
Don't trust that vacant expression.
Ooh, they're
There's
wily.
something going on behind those beady eyes.
Yeah, like I'm a velociraptor. Do you know that gull is sitting there, and in its mind it's just going, I'm a velociraptor. I'll eat your toes. Yes. And your ice cream cone and your blue chip packet. Oh my goodness. This is This Week in Science. Justin, what did you want to talk about?
Oh, it's that time again.
Yeah,
okay, so what did I have here? So the oldest synthetic material on the planet, a manufactured substance that didn't exist in nature, or was made by a multi-step process, we might associate with intelligence. And you can. This is researchers at Eberhard Karls University, Tübingen, Germany. They looked at birch tar in ancient Neanderthal tool. It was used as an adhesive to attach different tool parts together, from some samples that are 200,000 years old. And they wanted to know how they achieved making their birch tar. And basically what they did was they ran their own archaeological experiments where they extracted birch tar from wood with different methods. One of the methods that had been out there that had been proposed, that it was just be a natural fire, and then the birch tar leaks out onto to rocks, and then Neanderthals came by and just found the substance and scraped it off. So what they did is they tested, what a natural fire might look like, what cooking the wood in sort of a fire pit, and then various sorts of above ground with a hole underneath, and underground, and all these sorts of things. Basically what they came to is comparing the chemical analysis of the birch tar that was 200,000 years old with the 5 different methods they used to extract. The one that was closest was an underground, basically earth oven distillation process.
Whoa.
Now, this is sort of significant in a few ways. Cognitively, this is one of those things that, humans were supposed to be the first ones to have a multi-step process where unseen things are taking place, in this case, like an earth oven or underground. This theory of mind that has to sort of exist to explain this to another individual, the multi-step process that has to be involved, the sort of exacting setup of the process, because again, once it's going, you can't adjust it. You can't turn the wood or anything. It's out of sight. And apparently this all took place 100,000 years before current modern humans had done anything like this. The closest fire use for modification in current modern humans is in South Africa, about 40,000 years later, where humans were thought to have been heating rocks before knapping them, before turning them into tools. So 40,000 years after, Neanderthals were making synthetic materials through a distillation process in an earth oven. Humans heated rocks so they could smash better.
But would it necessarily need to be an earth oven, or could it have been, I don't know how some people, they do like a goat roast or a pig roast where they dig a big pit and they get coals in it, and it just is a slow heating process underground and not.
So the things that they could tell is that it wasn't above ground. It wasn't in just a fire pit or a natural fire situation, because there was low oxygen at the time of heating. That they could tell from their chemical analysis. There's a couple of methods that could have been utilized for doing it underground, sort of like a roast, like you're talking about. There was enough mineral infusion in the tar that they could tell it was in contact with earth. So it was a buried process, but the controlled oxygen in it, the low oxygen controlled environment, requires a fire to be able to continue to heat and do extraction without burning up anything. So,
is there any way to tell the difference with what they've found in their whole process of figuring out how this could have worked? As like a fire, as tool use, creating, using heat and creating the birch tar versus, there was a forest fire and they were able to scrape the tar, off of the birch trees as they wanted.
So the oxygen levels in the tar that they could tell that it was exposed to at the time of heating. The oxygen levels only work for underground. They did not match any of the above ground scenarios. The other one is there's no soot infusion.
Okay. That would
it was heated in a way that wasn't under the where the wood was not
burning
above the fire.
Yeah.
Or under the fire.
Yeah.
What am I saying? No, above the wood would not have been above the fire where the soot would have infused as the birch tar was coming out. So it was heated underground, likely with the fire on top of it. Yeah. So
fascinating.
A very specific process, which also then has to be, to communicate, teach, train, pass on an intricate manufacturing process like that. You also have to sort of have an idea of why you're attempting to achieve it. Now, I always have some, when they're thinking, this is so thought out and well understood. The other possibility, it's just windy.
Yeah.
It's just windy, and fires are bad when it's windy. So you dig holes for the fire, right? So you do stuff when you're going to do a prolonged fire. You might do it underground, and it would be hard to find that evidence
in a cave.
Well, the other thing is we talked about a while ago about the Neanderthal trophy hunting, where they had collected skulls,
right?
And then these skulls were also found in these caves around the area of the fire, which would have made them look really cool, right? So you have all these sort of interesting aspects of the way Neanderthals were utilizing fire in a much more complicated, intensive way.
Yeah.
Than
prior to humans. Yeah.
There's another study out that's talking about that looked at the cognitive differences between the way Neanderthals invented fire, which is they did the flint smashing, the striking to make the sparks fire, versus current modern human populations in Africa invented the whole drill and the dry wood and with the little string, and you pull and it makes the fire with that.
Friction, yeah.
And that's considered the technologically more advanced version, but it also might not be. I was thinking it might not travel as well in the Eurasian environment where it's damper and wetter. You can walk anywhere with your wet rocks, and once you're smacking them together, they're going to spark. Whereas if you had a couple of sticks with you and they got wet, you're doomed.
Yeah.
So also the cognitive dissonance in the fire making tool kits that they used.
Yeah.
Also actually kind of matched the environments really well. So
Very
it wasn't
much.
necessarily because of cognitive differences or abilities to conceive of things. It's maybe just accessibility of the materials to do it. And then you can also imagine Neanderthals accidentally inventing fire because they're constantly making the stone tools and knapping stones together. And then one sparks. They didn't have to go and I'll take a couple bones and smack them together. No, that didn't work. Well, let me take a piece of wood and this small pheasant and hit those together. No, that didn't work. They're experts at stone work, and so they could easily figure out which ones make the sparky things, and then, the technology goes from there.
It's And I've just learned this evening as a result of this study that apparently birch tar is used as not just an adhesive, also a sealant, and in medicines that it has antimicrobial properties as well, which is fascinating since we also know that the Neanderthals were potentially doing medicine type, they were healing each other if they could. And you can also buy birch tar essential oils because I guess that's a thing.
You can get any kind of essential oil.
Yeah, that's true.
There's
Extraction
also
technology.
birch tar soap for eczema. I have no idea about these things.
Okay.
And the antimicrobial thing is very important too, especially just if you're just using it as an adhesive or if you're weatherproofing your loincloth, whatever it is, because it's not going to get fungicides. I mean, or it's not going to get organically decompose on you. You know, it's not going to melt in the rain, in the dampness. It'll survive that, and it's not going to decompose and be ruined. So, anyway, just more evidence that Neanderthals were at least as intelligent. Actually, the story I didn't bring, which was about the napping versus the drill to make fire kits, they ultimately suggest that the cognitive abilities required to do those things, because modern humans and Neanderthals were separated for 500 to maybe 800,000 years.
Yeah.
Must pre-exist. We're very likely not convergent evolutions of cognitive ability, but more likely a sign that the longer, deeper history ancestor also was capable of communication and cultural passing on of knowledge and complex thought.
Well, animals do that.
Exactly.
Lots of animals do that.
Yeah, to a degree, yes. That's why we don't talk about tool use in terms of human intelligence anymore. Now we have manufacturing synthetic.
Right.
Now I got a whole other list. Where we
Who
stop using
manufactured
tools,
it
because
first?
any animal can use a tool. That's not a sign of intelligence, Blair. That's just what animals do.
Oh, my
Last
goodness.
story, talking about cultural education and being able to pass down knowledge. Marshall University study found that virtual sex education tool that they created or was created improved reproductive health knowledge scores and measures of self-efficacy among adolescent girls. This is published in the journal Sex Education. Researchers found that sexual health knowledge scores on a validated scale increased among participants, along with improved measures regarding of understanding of their birth control, healthy relationships, sexually transmitted infection prevention. So they kind of took a test before and after. This was conducted on females age 14 to 18. Participants answered questions about past experiences with school sex education programs. They then completed an online curriculum at www. This website, marshallteentalk.org is the site. Covers a range of sexual health topics rather concisely. It has some short animated videos. And then there's a post survey that reassesses participants' sexual health knowledge. And according to Jenny Yost, associate professor at the Marshall University School of Medicine, senior author of the study, adolescents use websites and social media for sexual health information. Therefore, there is a great need for accurate, evidence-based online reproductive health tools. Marshall Teen Talk was designed specifically to provide local adolescents an accessible and accurate resource for reproductive health that they can trust. This study validates the website as an effective teaching tool. So, that's awesome. I can see the value of a vetted sex educational tool that young people can access without having to sit in a class full of awkward peers or awkward gym teachers.
Right.
That's my question, though. Is this study measuring it against no sex ed? Or is this study measuring online education versus in-person sex ed?
So that's what caught my attention. Because 30.3% of participants reported that they had never had sex education classes in school. Now, I'm just a liberal country boy from a rural university farm town in California. But I thought sex education was a mandatory part of school because I had multiple classes growing up.
Well, it depends if you're public or private. It also depends if your parent has signed a waiver to remove you from that class, which all parents have the opportunity to do.
Yep.
And it also depends on the laws in the state that you live.
So,
or
very
the school,
important.
Or county school district.
Because I was wondering, where in modern America, first world developed country America, is 30% of people 14 to 18 not receiving sex education?
Yeah, that's a thing.
Where is that possible? And
America is where.
So, well, the answer, because it turns out this is where Marshall University is. The answer is West Virginia. Is a place in America in 2022.
One of the places.
Is a place where young people are being severely undereducated. So, in a place like that especially, but also elsewhere, I guess, where people have questions or interests that they're educational, want their information in a straightforward, trustworthy online source, I can see how this would be extremely helpful. But this might not be it. Because one thing that you will not find discussed anywhere in the conversation or the education about STIs or birth control options or any of this is there won't be any discussion of abortion. And the reason there won't be any, even in the informational resource links to abortion, is because abortion is completely banned in West Virginia as of September 2022.
Yeah.
Marshall being in West Virginia is likely just adhering to local law.
Information. Yeah.
There's 1 minute that discusses LGBTQ orientation. It's not negative. It's not really that informative either. But there is at least a link in the resources to Advocates for Youth that may lead to a path to a future where more information is accessible. But no mention of abortion. So while the website could be a good resource for the 30% who are receiving no education in West Virginia,
they're still not
seems like
getting info on that.
Local law has got it censored. And then the next thought I have is, Tennessee did away with sex education. They also have the highest rate of teen pregnancy in the nation. Wow, what? I didn't know. But what if so is this resource that they've created to reach rural youths who aren't getting sex education then can also have to remove stuff if a law is passed? Censoring information.
It depends. I mean, there's really interesting stuff going on in relation to, and I'm going to connect this in, I promise, to the TikTok ban in Montana. Because if you can find a state where there aren't all of these restrictions and you publish an online sex ed course that's authored in there, like in California, for example, but it is accessible to all 50 states. Anyone can Google it and watch it.
Right.
That's great. There are weird things going on right now related to exactly what you're talking about. When there are there are state-specific laws that prohibit or censor specific internet content. So this is really the question I think that we will see. This is where science and politics mesh, is like, can you impact access to information? Can you have a sex ed course that covers things like abortion and LGBTQ issues that anyone can access? Or can you put firewalls in certain locations that prevent that information from being accessed?
Yeah, certainly from schools, from private internet networks. Those firewalls can be put into place. Much harder to do on a statewide, larger level. But I think that is a huge very important question to be asked. The information could always be out there. And there are whisper networks already for, various people put together informational networks and resources and get the word around, but it just makes it a lot harder to access.
Yeah, and so the point, I guess, what I came to is I applaud the idea of this.
Yeah.
Why is it in West Virginia?
Well,
because they have a need, right? Because
they have a need. I get it, but this needs to be in a state where you aren't having to censor your information if your information is meant to get to.
Yeah, and I'm gonna guess this is not the only place that this kind of information is made available. Where there are many more websites in many other places, put together by different organizations, different universities. This is just we've learned about it because they published the findings related to how well their online tool worked. So they've done a study and they published it. And so that's why we know about this one in particular. And so it's great for us to be able to talk about it in the sense that this is in West Virginia and what else is going on there. That in other states where things are more accessible, that this tool is, or tools like it, are probably available, accessible, but maybe the information has already been more available in the first place. And I think comparatively across different tools and different states and different areas, I think that would be an interesting study to see. What are we going to find when we look comparatively?
And I think also this is a reminder to parents of children that are entering an age where they need to learn about these sorts of things, to know what your school district is teaching, know what your state allows in the curriculum, and if you don't agree with that, to find alternative resources to provide to your child, because in the current day and age that we are living through, you cannot count on the government to teach your child the complete sex ed story. You have to find out if that's happening on your own. You
have to know what your local government is if politics are getting in the way of health information. And mostly, I'm also disappointed because this is a perfect website for a father with daughters. Like, it's time to have that talk. Here's the website. I'll be back in an hour. 45, 2.5 hours. I'm going to actually be on vacation. You can ask your mom any follow-up. There's tough things in communication, which is, and also, I can't leave it to myself. I'm going to get it wrong. I'm going to get
That's the thing. You can't count on just parents to know the most up-to-date information.
Yeah.
Those of us who went through sex ed decades ago may not have the exact right information to provide to our children.
Right. And the reality is, we love to think that we are the teachers, the ones most responsible for all the information that our children are coming to use in their lives. But really, more often, they're relying on their peers and on the educational surroundings and social media, the internet, all that kind of stuff. But we wouldn't want to second guess ourselves here about all this too much. Or would we?
I'm always second guessing myself.
And that could be genetic, Blair.
Oh no! Aren't you mom and dad?
And ancestors going back even further to I don't know how far back, because this study looked at mice. So it could have implications for people. But these researchers were supported by a grant from the National Institutes of Health, and their published study was called ARC Regulates a Second-Guessing Cognitive Bias During Naturalistic Foraging Through Effects on Discrete Behavior Models. They looked at 1,609 foraging excursions by mice. And normally you think, okay, the mice are going to go out and they're just going to look around and they're going to be like, oh, look, there's some food. I like
Mhm.
that food. I'm going to eat a little bit of it. That's great. Maybe I'll take some of it back to my little home base, whatever. I'll go back to that spot. Oh, there's still food there. I'm going to go to a new spot. Oh, look around. They found that the mice that they were looking at as they foraged in this naturalistic laboratory setting, of course, is not a real setting, but they found that they had 24 behavioral sequences that they did, like building blocks, over and over again. That they had specific sequences of behavior that were used in their foraging. And the mice foraged and kind of put the sequences of behaviors together. And like in between the different sequences that were hardwired, they'd do something spontaneous, like go a different direction. But then back onto their foraging sequence. That's the next foraging sequence that they're going to do. And together, they found that they had never seen this before or determined that there were these repeated behavioral patterns because of the way that there was a spontaneous behavior. It made it seem very, very complex from the outside. But what they found is that one of these behavior patterns was a second guessing behavior. And the researchers found that there's a gene that we've talked about before called ARC, A-R-C. ARC is known to be involved in learning and memory. And there are many interesting research paths specifically focused on this protein or the protein that the ARC gene produces. They got rid of the ARC gene, and 6 of those 24 not spontaneous, but stereotyped behavior sequences got shifted, and it short-circuited a second-guessing behavior. Like, oh, I've seen food here before. Maybe I'm going to go here. Nah, no. I don't want to go there. I'm going to go someplace else. So these mice that had originally been second-guessing themselves about where they wanted to go and find food, suddenly they weren't second-guessing themselves
They
anymore.
followed through with their original plan.
They followed through with the original plan. Exactly.
Does that mean that they stopped the little spontaneous interludes?
They were different. No, the spontaneous interludes were a little bit different. So, but what they think is that everything else, the memory, even though they altered ARC, they got rid of it. The mouse memory still seemed to be fine, and learning still seemed to be fine. It was just the second guessing of what they were going to do next was the thing that disappeared. Right. So I guess in a sense, you could say that spontaneity is something that was also short-circuited, because instead of following through to like the next obvious place to go look for food because you've seen it there before already, that maybe a mouse would be like, do I go left? Do I go right? Do I go left? Do I go right? Do I go where I know there's food already? Do I go someplace where I don't know there's food? Now the mice wouldn't do the spontaneous choice. They would just do what they already knew. But the researchers say that what they think this is that this second guessing is possibly partially genetic, and that the decisions that we have second guessing because it adds more novelty, which could allow for more exploration, and it could have been advantageous to mice in the wild, or the animals in the wild, to not necessarily go with the sure thing, but to second guess and to mark the other circle on the multi-choice test.
So this is right now when I wish I had time to go and try to research something on the fly. Now I want to know right now if people who have no internal monologue have a different ARC gene.
That could be very interesting.
Because that's where my second guessing comes from. It's all internal dialogue. It's all I'm going and some little voice imagine. You keep saying you want to go left instead of right. Why don't you try it today? Oh, okay. It's internal, but without the internal dialogue, I don't know how I would second guess anything. How would you even? I don't even know how you do a first guess, but second guessing definitely is off the table.
Right. So the first guess is that, you've gone and you've found food somewhere before. So there's just no choices. There is no, do we have Chinese or Mexican for dinner tonight? There's just Chinese because that is the only thing. Yeah. And there's no, second guessing or, I'm not going to go into other food-based stuff because, anyway.
It's good enough analogy.
It's good enough analogy. Yeah. But you don't, the second guessing is, I think if there's no other option, why do you have to have an internal monologue?
Yeah. I'm insanely curious about people without an internal monologue as it is, but now I want to see if the ARC gene is involved.
Huh.
Yeah.
That would be interesting. It'd be interesting to see all the different kind of connections of what's going on in the brain with the ARC gene. Because you're right. I feel like second guessing is a really complex behavior.
Very complex. Yeah.
And so they've simplified it on there.
In this particular study, it's, very simplified. It was like, oh, look, mice don't always just go to the spot where they found food. They go to these other patches that don't necessarily have food that they've, or that they know might be empty. Where are they going back there? Like what's happening, Why are they second guessing their choice when they know there's food someplace, but there may or may not be food someplace else? Why are they second guessing it? We know the ARC gene is involved in memory. So are they just is it a faulty memory? Like maybe there's some food left, or like maybe sometimes because of different seasons and different plants that,
I've got a suggestion.
Grow more. I don't know.
I got a suggestion there too. Food very likely where you just found it, but the other spot seems to have a competition to it. It's the blue chip all over again. I'm gonna go to the contested food spot just because that's the one that you gotta be there at the right time for. The other one will be there. It's fine. I can get that anytime. But the other one might be time dependent or competition dependent, and so I should go take a look.
I was thinking it's more like a gambling behavior. Yes. Like you're taking a risky move, right?
Right. Known reward or...
Yes.
Is there maybe?
Yeah. Do I cash out or do I put it back into the machine? Do I go again?
Put it all on black.
Yes.
Yeah, this is all very interesting. And yes, it's studying mice, and maybe one day we'll understand this second guessing food choicing of mice, but it could also help us if we can figure out how the ARC gene is working more specifically within mice. Maybe it'll give us some insights into things like gambling, into our second guessing, into how we live our lives. And I don't know. From now on, though, whenever I'm second guessing, I'm going to say this is totally just because, this is evolutionarily advantageous. Whatever choice I make.
There you go.
Whatever choice I make, it's fine. It's good. And then final story. Blair especially, but everyone, according to a new study out of the Public Library of Science 1+1, published today, May 31st. Researchers out of UC San Diego and out of the Max Planck Institute for Evolutionary Anthropology in Germany have found out that kids, not goats, human children, are more curious than other great apes. They compared 2 to 3-year-olds against gorillas and chimpanzees and bonobos and orangutans on a little task in which they had 2 cups. And the 2 cups had grapes. One of the cups had more grapes than the other cup. Now, for the great apes, grapes were awesome. For the kids, they had to switch it up and they got stickers instead of grapes, because apparently I'm sorry, and I said 2 to 3 years old. It was aged 3 to 5 years old. Oh, okay. But apparently 3 to 5 year old humans like stickers more than they like
love
grapes.
Stickers. Yeah.
And I don't know whether this is because the adults who put this study together were afraid of the kids choking on grapes, or whether the kids really like stickers more than they like grapes. But anyway, that's just one little thing in this study. One cup was transparent. The transparent cup had fewer of whatever the reward was than an opaque cup that was right next to it. And over and over and over again, this is basically gambling, right? It's the certain choice of a small amount of grapes or stickers, or what is in that cup I cannot see into? Yeah. Do I choose it?
Do I invest in a savings bond, or do I bet it all on the ponies?
Exactly. And over again, the apes bet on the safe choice, while the kids, up to 83% of the time, they went for the cup they could not see. Kids, over and over and over, were like, I just have to see behind door number 3, Alex.
Man, that's tough, though.
You know what they're thinking? You know what they're thinking, though? Oh, I really hope there's grapes in there.
So this is my problem with this.
Can't eat stickers.
These animals are betting on something vital to survival, food.
But I mean, they're treats.
They're treats, but it's a different value item because it has caloric qualities.
And and of course these are not wild apes that they've gone out into the wild and done this. These are zoo apes. These are apes that exist in habitats where they're very well fed. So if we'll think about this, like from the arguments we've had over and over again about the marshmallow test,
right?
This is, these are apes that are very well taken care of. They are not afraid of starving.
Right.
I feel like
it's tough because I also feel like human children, generally speaking, do not have to fend for themselves to eat.
Oh, I did. Oh boy, I did. Oh my goodness, let me tell you this.
Generally speaking,
3 to 5.
3 to 5 year olds, right? And it's also so that's the other problem. Like, stickers are just for fun. It's just fun. So you'd be like, what could be in there? A different toy? More stickers? Less stickers? I don't know. I want to know what's in there.
I know. I can't see what's in the cup. I have no idea. But yes.
Listen, I'm sure maybe kids had many reasons for doing what they did. It just, look on the outside looking in, it seems like a false equivalency to me, based on the value, based on the quality, based on what there is to win or lose out of the deal. Yeah,
it's also it could also be a very like, what do kids like? I don't know. They like stickers. Well, I put those in there. Kids go crazy for them. Do they really? Oh yes. And then the kid's looking at a bunch of stickers, going, "That's not fun enough. That's not interesting enough."
Yeah, why didn't they use marshmallows or chocolate chips or a food item at least?
Anything other than a boring sticker. Yeah, well, give them gold star stickers. I used to like that as a kid. Knock it off.
So the last phase of the study. So the last phase of the study for all of the chimps and kids, they gave them an idea that there's just more of whatever is in the transparent cup in the opaque cup. Basically, we're like, look, you could see everything, and the children kept engaging in some aspect of exploration to get their choices. And the great apes, once they knew there were more grapes in that opaque cup, they switched their strategy, and they were no longer risk averse, and they only went after the opaque cup. So it's so I think the risk aversion aspect and what you were talking about there with like survival based reward, or what is there anyway. The authors are like, well, of course, this means humans generally are, going to be more exploratory and less risk averse. And of course, that's why we're so successful. No, that's not what they said. But anyway,
well,
that's not what they said.
So the other, The other thing is human intelligence and human environments, especially now, are so much more intricate. There's so much more to explore. I mean, if you had too much curiosity as a young gorilla, I imagine you would go insane with boredom. Because there's only so many leaves to turn over. There's only so many sticks to...
You would most likely end up dead.
Justin, you think the jungle is boring? Is that what you're telling me right now? Like, what?
Compared so many ants to watch.
Compared to the level that of intricate novel things that a human child can interact with in a day and still be hungry for more, yeah, I think a jungle.
Oh wow, I think you are in the wrong universe on this.
I think a jungle can be very boring. How much time have you spent in a jungle without cell service where you're like, yeah, I've seen enough jungle? I feel like it would take me maybe a day or two not being from the jungle.
Wow.
But
no, I would be spending my time trying not to get killed. That would be pretty much what I would.
Oh, you left the tent. See, there's the difference between you and me. You actually went out into the day, stayed.
What you're describing is the enrichment we put in enclosures for animals to replicate the
wild
world
scenario.
Like that's what we do to ourselves. That's what we do our children. We enrich our children's lives because it is boring compared to the wild. That's what you're doing.
Yeah. Maybe.
You are stimulating behaviors and mental sensations that nature did before we domesticated ourselves and put ourselves in boxes.
Yeah.
That's what you're doing.
Yeah.
And there's definitely still exploration that's happening, but the question is, why is it a higher percentage of human children who are more likely to engage in the exploration in this particular study than the great apes? Yeah. And is this something that is really a difference between our primate relatives and the human brain? And is this curiosity and exploration one aspect of our development as humans that is really different? And I think that's a question that deserves a lot more study.
Yeah,
not
if
whether or not
that is what they tested. Let's just say that is what they tested and that is what they found. I don't think that's a result of us being more advanced or any of these other things. I think you hit the nail on the head talking about trying to not die in the jungle. We don't have selective pressures to prevent toddlers from risky behaviors anymore because by and large, we keep them alive.
You attach the bookshelf to the wall so they can't fall over. We don't feed them grapes unless they've been sliced so that they don't choke on the grapes.
Oh, yeah.
Exactly. So we're not selecting out for ultra risky behavior anymore. So really all that's happening is like adaptive radiation where we're allowing risky behavior to happen untethered because there's no pressure on that behavior.
Except that I would go into the deep well of human history and look at all of the diversity of survival strategies that humans have and environments that humans have adapted to. And I would compare that to gorillas who have the last bit of their rainforest goes away are gone from the planet. I mean, they're going to go panda bear in terms of I need this and nothing else.
But you're also, I think you're changing, adapting to an environment versus adapting an environment to you, which is what we by and large.
No. If you're going back into, I'm not talking about current modern human history. I'm talking about going way back where you would have a fishing subsistence versus a forest gathering subsistence versus hunter-gatherers who are hunting different prey. I mean, humans.
Well, that's the question. How would those toddlers do on this test? We don't know.
Which we can't test.
I feel like they'd be fine. But I think that humans' explorative nature is part of the human intelligence that gained all this knowledge by experimenting with that cup that was a mystery. And I don't think that is
why science
an animal
is so awesome.
intelligence I think you're going to lose on the comparative animal intelligence. They're just as curious about everything, and they're just as exploratory, and they're just as... I don't think so.
But the reason
And
is
so
the question is why. And so what are and in the last sentence of the abstract that the researchers have put in there, I mean, they are still arguing that these differences are definitely there, but they say they mostly lay in motivational dispositions to explore the unknown. So what are the motivations for exploration? Is it real, like cognitive brain differences, or is it survival and risk aversion, or maybe humans have started adapting to these safer environments where they can be more exploratory? There are so many things that we do not know.
And there I would totally 100% agree with Blair, because I think one is food and the other is a resource that you can get anytime you want. If you're a kid, you can get stickers.
There's no stakes.
There's no stakes. Yeah, totally agree.
Yeah. Explore. Stickers. I got 5 of those at home.
Yeah.
I mean, 5 sticker books.
Let me see what else you got.
What else do you have?
And if I cry, I'll get those anyway. I'm gonna get them. Like I'm gonna whine my way into the treat if all else fails.
I hope nobody's gonna cry right now, because I think we are all out of stickers. The science sticker cup is empty for the night. Yes. Mm-hmm. And we've made it all the way through the show. Thank you, everyone, for being here for another episode of This Week in Science. I would love to send some shout outs to our chatters. Everyone in the chat room, thank you for joining us and chatting. Definitely thank you to Gord and Arnlore and others who helped keep those chat rooms very wonderful places to be. Fada, thank you so much for your help with show notes and social media. Awesome that you help with that. Identity 4, thank you for recording the show. And Rachel, thank you for your editing work. And of course, as always, I cannot forget, I must thank our Patreon sponsors. Thank you to Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Kent Northcote, Rick Loveman, George Corris, Pierre Velazarb, John Ratnaswamy, Carl Kornfeld, Chris Wozniak, Vegard Shefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan, Don Mundus, Stephen Albaran, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredis 104, Sky, Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, David E. Youngblood, Sean Clarence, Lamb, John McKee, Greg Riley, Mark Hessenplo, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Johnny Gridley, Remy Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, who's on a boat. Please don't get attacked by orcas. John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Adam Mishkan, Kevin Parachan, Aaron Luthen. Oh, and by the way, happy birthday, Adam Mishkan, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, and Tony Steele. Thank you for all of your support. And if you would like to support us on Patreon, please head over to twist.org and click on the Patreon link. On next week's show,
we will be back Wednesday, 8 p.m. Pacific time, or Thursday, 5 a.m. Central European time. Pick your show, either one.
It's the same thing.
Broadcasting live from our YouTube and Facebook channels and from twist.org/live.
Hey, do you want to listen to us as a podcast? Perhaps while you sequence your genome? I don't know. Just search for This Week in Science if our podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes, links to the stories will be available on our website, www.twist.org.
You can also contact us directly. Email Kiki at Kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be spam filtered into an opaque cup that all of us are too risk averse to look inside. I'm so sorry.
Sorry about that.
You can also hit us up on the Twitter, where we are at Twist Science, at Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
It's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.